Good afternoon, everyone. This has been a session that we've been waiting for months with these guys. Why? Because we're gonna talk about a project that we do together, and we are super proud at the ETPN to be part of this project. So normally, when I participate in project for the ETPN, there's two situations: whether I understand very well the project and I'm super excited, whether I don't understand anything about the project at the beginning, and I'm also excited in a different way because I know that I will have to work hard. So this project is METRINO. METRINO is an Euramet project. Do you know Euramet by chance, or absolutely not? Yes, you know. Euramet is all about, let's say, funding for metrology. Metrology being the art of precisely measuring things. And indeed, in nanomedicine, we need to measure lots of things with lots of methods. And we need standards, and we need to know how to do it not only for the clinic, but also on your daily practice at the lab. So the whole purpose of METRINO is somehow to simplify your life, if possible. But it's not simple to simplify the life of others, right? So this group of experts is really working really hard for now a year and a half, a bit more. We have 3 years of funding. We are already thinking about the future of METRINO, but we decide that, okay, webinars are great, sending questionnaire, you have always the same motivated people that answers, never the clinicians. I'm not saying they are not motivated, I'm just saying they don't answer to Google Forms. I mean, really, they don't. I don't know why. Should send a fax, maybe. But the idea is really we thought, okay, Nanomed Europe will be a big gathering of stakeholders in nanomedicine. And instead of doing yet webinars and webinars, let's have a room and we make a workshop. Not just a session, but we called it a workshop because we would like you to raise your hands, to ask questions, and to talk about your needs, like on your daily basis at the lab. What is the thing that you would really need in terms of standardization that would really help you? And it can be also things that you would like to stop doing. I remember really well, the first time we have a workshop, it was 2 years ago in Liverpool. It was the beginning of METRINO. You know, the first day of METRINO, it was with Fanny Caputo, the ex-coordinator. And I remember that some students said, we would love also to have a list of things that we should stop to do in characterization, things that doesn't work. Protocols that are wrong, you know, and I won't quote any methods for size distribution. No, I'm not looking at anyone. I'm just saying that these are things that maybe we should stop to do. I'm not sure we're going to solve all these problems today, but at least we have 4 fantastic experts that will talk about METRINO, and then we have a panel, let's call it an open discussion with you. We want you to talk because we'll transform your ideas today in priorities for the last part of the project, right? Like for instance, is it about webinar? Is it about really practical guidance? We don't know, and we want you to tell us. But enough of a long introduction for a short session, and I would like to welcome on stage Robin Schürmann from PTB, who's the coordinator of the METRINO project. Welcome.
Yes, thank you very much for the introduction. It's an honor for me to be here and to present the METRINO project and to tell you what metrology can do for nanomedicine. And I'm really looking forward to the discussion with you that we make the project to have some real impact on your work. You all are like expert in nanomedicine, and I see also some expert in metrology in here, but that nobody get lost during the next session, I will briefly explain you what metrology is. So metrology is the science of measurements and its applications. This comprises like all theoretical and practical aspects of a measurement, like it doesn't matter what kind whatever you measure and what's the uncertainty of the measurement. And it's about providing reliable, recognized measurements which are traceable to a common reference. And the best common reference typically is the SI system of units. And the traceability means that we have an unbroken chain of comparison which links the result of your measurements to your reference. And together with standardization and accreditation, metrology is one of the key parts in the quality infrastructure. What is how is metrology relevant for nanomedicine? On the one side, there are you, like ex-scientists, small companies, industry, having great concepts and nanomedical applications which they want to bring to the clinical practice. But to bring your solution to the clinical practice, you need to meet the requirements of the regulation and provide that the safety, quality, and efficiency of your product. And but to show the quality of your product, you need like a lot of complex characterization of your particles, and moreover, you don't have any reference materials, you don't have good standardized test materials, and there METRINO wants to support you in the preclinical translation of your work by providing reliable standard protocols and reference materials which support you in the characterization of your nanomaterials and to answer the regulatory questions. METRINO is an European Partnership on Metrology project. It started in June 2 years ago, and it has 20 partners, which are metrology institutes, universities, research and technology organizations. We have one hospital, different pharmaceutical companies, and also the ETPN as partners. And the main pillars of METRINO are, on the one hand, the development and harmonization of so-called standard operation procedures for the characterization of the critical quality attributes of your nanomedical products and to make these SOPs available to the community. The second pillar is to shape a roadmap for the development of reference materials, namely lipid nanoparticles, liposomes, and also metal oxide nanoparticles, which are used in cancer therapy as radio enhancer or as contrast agents in magnetic resonance imaging. And there we are working together with partners from industry that we make sure that the materials we develop represent the formulations on the market. The third big pillar is the community building. We want to establish a collaborative network which brings together scientists, metrologists, to get to the standard development organizations, the regulators, and the industry. How does this community building look like? We organized a workshop together with Nanomeasure France, which was called the International Standardization Roadmap for Nanomedicine. The results of these workshops are also published in Drug Delivery and Translational Research. And there we brought together the key players in nanomedicine characterization and standardization. And the aim of all participants is to harmonize test methods for nanomedicine that address the net regulatory needs. And it turns out that the landscape for standardization in nanomedicine is very fragmented, and there's a need for a unified approach. Let's have a look at this fragmented landscape. If we're looking at nanotechnology-based products, we have on the one hand organizations looking at the product quality and safety, like the regulators for chemistry, food, drugs, and medical devices. We have institutions like the EDQM, the European Directorate for Quality in Medicine, which is looking at the quality. We have guidelines, for example, by the OECD, which is providing test guidelines. And on the other hand, we have these measurement methods for the critical quality attributes. There we have the method development, which is done by researchers, by instrument manufacturers, and by Metrology Institute. We have the part of method validations, for example, in the pre-standardization framework VARMAS, and we have the standardization organizations like ISO and CEN. Let's have a closer look at the standardization and validation process. On the one side, we have the metrology. If we want to validate a method, for example, to measure the diameter of spherical nanoparticles, at the Metrology Institute, we have a reference methods. These reference methods allow us to measure the diameter of a particle traceable to the SI system of units. With these reference methods, we can certify a certain reference material with the value for the diameter of these particles. When we have harmonized methods or standard operation procedures or test guideline which we want to validate, which is measuring the diameter of the particles, we use these reference materials and we perform an interlaboratory comparison with expert laboratories and the National Metrology Institute. And this way we can assess the trueness of the methods and also identify some bias of the methods. And if the values of the methods between the different institutes match the true values of the particles, then we have validated the methods. From there, we can go on to the standardization. If we have like a standard, this allows proficiency testing, which is validating the skills of your laboratory that you are capable to measure this parameter. And this is an important step in the accreditation of laboratories. What we are doing practically, we are developing reference materials. Like the ISO definition for reference material this is a material which is sufficiently homogeneous and stable with one or more specified properties which is established first for purpose for intended use in the measurement process. And there we are working on superparamagnetic iron oxide nanoparticles, as they are used as contrast agents, hafnium oxide nanoparticles for cancer therapy. We have lipid nanoparticles loaded with RNA, liposomes, which is like the biggest group of available nanomedicine products, and which is not a typical nanomedicine product, is like fluorescent upconversion nanoparticles. Why are we working also with these particles? These particles are able to be measured like with fluorescent methods, mass spectrometry methods, with a huge toolbox of methods, and this enables us to correlate different microscopic techniques, and there we also need materials with defined properties for this purpose. Then we are developing standard operation procedures. There we are working on the critical quality attributes on the one hand of metal oxide nanoparticles. There we look at the size, degree of aggregation, and number concentrations. Typically you think we have 2025. Why should we measure the size of like nanoparticles? But if you look at this typical superparamagnetic nanoparticles, they are super small, below 10 nanometers, and they to have their like function, they need to form like non-regular aggregates. So we have like samples which are huge polydispersity, different shapes, different sizes, consisted of different core particles, and this is like really a challenge to identify the critical quality attributes of like such materials. Then we are working on the coating of the nanoparticles, and besides that, we are the second focus is on lipid nanoparticles. There we are interested in the structural integrity and lipid chemical composition. And here you see that we are using ensemble methods, where we are measuring a quite big ensemble of nanoparticles with, for example, small angle X-ray scattering. But we also use particle by particle methods like cryo-TEM. There we have a special focus on the cryopreparation methods, because with the cryopreparation, we open up the whole toolbox of like surface science analytical methods to our lipid nanoparticles. Then the regulators are interested in what happens when you bring your nanoparticles in biological media. How is the stability in, for example, blood? How is the biotransformation in this media? And there we have developed protocols for sample preparation, and we are working on fractionation protocols to separate the particles from the biological matrices. Currently, we are working on an ILC, which we will see in another talk on flow field fractionation of lipid nanoparticles. There we are working with different institutes from the project together with collaborators from industry. In an ILC you send around reference material candidates with SOP. These are measured by all the different laboratories, and then we compare the different results, how the institute can like work with the SOP, and this will provide input to some ongoing standardization activities. last but not least, regulators are interested in the distribution of nanoparticles in biological tissues, and there we are working on correlative methods to localize and characterize and quantify the nanoparticle uptakes in tissues. There we are preparing different kinds of model tissues with different relevant nanoparticles, and we have like a whole battery of different advanced microscopic techniques. And with these techniques this year we want to initiate an ILC to study the nanoparticle uptake in biological tissues. How can you get in touch with the METRINO project? On the one hand, we have like collaboration with in the scope of interlaboratory comparison, but also to test the standard operation procedures we are developing and the reference material candidates. Currently, we have like 7 ongoing collaborations. Then we have stakeholder advisory boards where we discuss the progress of our project with expert in nanomedicine, which we should exploit a little bit more than we're currently doing. And to keep updated on like webinars or workshops from the METRINO project, you can visit our website, metrino.eu, or follow us on social media to be informed on the updates of the project. Yes. Thank you for your attention.
Thanks a lot, Robin. Very nice overview and introduction. We'll keep the questions for after, so if you have questions, please don't forget them. You have notes on your iPhone, right? And we'll move on to the second speaker. She's Elina Andresen, and she's coming from BAM. And I would not dare to say BAM in its full name in German because it's a bit complex. It is a bit complex. But Elina will explain everything that we do in METRINO towards development of reference test materials for metrology in nanomedicine. Nope.
It is not
exactly.
opened.
It's supposed to be open.
No, it is
It's not open. Okay. I said supposed. Huh?
But my French is not that.
Okay. Alors.
Okay, good afternoon also from my side. I'm Elina Andresen from the Federal Institute for Material Research and Testing in Germany, also just short BAM. And in the METRINO project, we are leading the work package 1, in which we aim to develop nanoparticles as reference candidates for evaluating, developing and harmonizing measuring techniques for biomedically relevant materials. So I would like to start with some definitions on the different types of reference materials. So the gold standard, the highest level of reference material, is the certified reference material. And this is a reference material with a certified value with a stated traceability and uncertainty. And such materials need to be characterized by a meteorologically valid procedures for one or more specified properties, and then they come with a certificate that provides the value for the specified property and all the associated uncertainties. And only such materials can be used for providing SI traceability of the measurement. If we skip the traceability part, we will just end up with a reference material. This is still a very homogeneous and stable sample for one or a set of specified properties. Only such types of materials, so certified reference materials and reference materials, can be used for assessing method accuracy and trueness. However, when thinking about toxicity testing, such homogeneity, as I will present just in a minute, for reference material is either ethically not acceptable, not feasible, or sometimes even not needed. Therefore, the NRC came with the term of a reference test material. So this is a material which is not tested for homogeneity and stability for one defined property. And when we take this term in context of the measurement of physical chemical properties, we refer to such samples as quality control samples for material assessed in interlaboratory comparison, and the homogeneity and stability of these materials is determined for one property, but then we speak for fit of purpose materials, for example, for different testing programs and ILCs. The increasing application of engineered nanomaterials requires reliable, validated, and in the best case, standardized characterization methods for the application relevant physical-chemical key properties as the size, the composition, the concentration, or the surface charge and the agglomeration of the engineered nanomaterials in different matrices. Most of these key properties we are addressing and studying within the METRINO project, with a small focus also on the surface group analysis. Here we have also a partner project which is putting more emphasis on this, on the surface group analysis. When having a look on the land map of different available nano-reference materials for life sciences and medicine, there are not that many. But there is an increasing need of such materials, and NRC addressed this issue and studied 6 different clinically relevant formulations, so liposomes and lipids with different charge. The measurement was is the size and the polydispersity of the lipidic and liposome formulations. And meanwhile, 2 of the materials are already available as certified reference materials. But in METRINO, we would like to contribute to filling the gaps and to provide more materials for as reference test materials for nanomedicine, and also for the development of different standardized operating procedures. So we hope we can fill the gaps. And therefore, we started with a very big selection of different inorganic nanoparticles and soft nanoparticles, so lipid and liposome particles provided by different partners in the groups. And for today, I would like to present you the reference material development that we make at BAM for our in-house made particles, the iron oxide nanoparticles prepared in a hot temperature synthesis, and also the multi-element nanoparticles that we like to refer to as upconverting nanoparticles. A brief introduction on these 2 types of particles. So the upconverting nanoparticles, as the name already says, have the ability to convert low energy excitation irradiation into higher energy visible emitted light. And this property is giving them some very unique optical properties, as the zero autofluorescence, so background free measurements. They also have narrow emission bands, and they are very resistant against photobleaching. And this property is given by the lanthanide ions, which are doped in mainly sodium yttrium fluoride host lattice. And these lanthanide ions make such particles also very interesting and easily detectable by a multitude of analytical and imaging methods, optical methods, as well as fluorescence methods and mass spectrometry methods. On the other hand, we are developing also iron oxide nanoparticles, which have a much broader medical application in MRE or cell separation and also drug delivery. So in order to prepare a reference test material, there are a few steps that need to be performed, starting with the synthesis and basic characterization of the material. In the METRINO project, we are developing citrate capped upconverting and iron oxide nanoparticles as test samples for size and concentration measurements. After a preliminary basic pre-characterization of the material, we have the bottling step, followed by the homogeneity and the stability studies, and after that we will provide our material to 3 SACS groups involved in the project in a small intercollaboratory comparison, which will be then the measurement that we will also provide for our materials. Finally, these particles would be also a cytotoxicity evaluation would be performed in the university by the University of Magdeburg. In the second part of the project, such materials would be also used for development of methods to measure nanoparticle uptake and distribution in single cells and tissues. Also here, different steps need to be performed, and at the end we have different methods for the tracking of the nanoparticles in tissue engineered models. How do we synthesize our materials? So we, for both materials, we use the high temperature thermal decomposition method. This means that we start with the precursors as oleates, and then in hot boiling solvents we perform the nucleation of the particles so that we end up with highly monodispersed particles. In the case of the iron oxide of around 10 nanometers, and for the UCMPs we have 2 different batches. Here just presented the 20 nanometers, but we can also prepare smaller particles. Of course, such oleic acid capped particles are not water dispersible and do not have many application fields. Therefore, ligand exchange needs to be performed, and here we chose the citrate coating as a biocompatible coating to make our particles hydrophilic. Next, the pre-characterization of the materials is performed. We use 3 different sizing techniques: electron microscopy, dynamic light scattering, and small angle X-ray scattering to determine the size of our particles. Then the surface is characterized so by a zeta potential, and then we use HPLC to determine the amount of citrate on the surface of the material, as this is important for interaction with matrices biomolecules. And also we measure the weight loss by thermogravimetric analysis. So we measure the weight percentage of organic ligands on the surface of our particles to determine the exact mass of core nanoparticles, which is later relevant for the calculation of the particle number concentration. Then the composition of the particles is determined in case of the UCMPs, which are very sensitive in ICP, OES techniques. We use this one, and for the iron oxide particles, we perform optical assay to measure the iron concentration, which is also relevant later for particle number concentration determination. The next step is then the bottling of the material. Okay, yeah. We prepare for both materials 100 to 150 equal vials with a concentration of 5 milligram per milliliter, volume of 1 milliliter. So now we have enough material. So this is from one single batch of each of the particle types. And this material we need to test in the next step for homogeneity. According to the ISO norm, the homogeneity testing should be performed. So the number of vials needed for homogeneity of a reference material is the third root of the number of units, but not less than 10. So if you start with less than 1,000 units, you need to perform the homogeneity with 10 vials. This is also what we did. The method was Mo-angle X-ray scattering performed at the CEA in Paris. And we measure always 2 aliquots per vial to have the in-bottle homogeneity, and then 10 bottles to have the bottle-to-bottle homogeneity of our materials. And the results are very nice. So our material, after the bottling, is homogeneous, and we can continue with the stability testing. For the stability testing of a reference material, there are 2 types of studies that can be performed. The long-term stability study, in this case, the reference material is just kept at a temperature. Here for the iron oxide, this is just room temperature. And then after defined time periods, the measurement is repeated, and then we need to check if the key property that we have defined is stable. But we can also perform so-called isochronous studies. These are accelerated stability studies, which also help to predict the stability of the material during transportation. In such an isochronous study, you will divide the different units, the vials of the reference material at different temperature, thereby storing 2 reference samples at the temperature where the sample is known or assumed to be stable, then samples at higher temperature and at lower temperature. And after defined time periods, the samples from the higher or the lower temperature are just put back to reference temperature and stored at this temperature until the end of the stability study period, and then measured to check which influence changes in the surrounding temperature will have on the particle stability. So to summarize what we are doing at BAM to develop reference material for the different techniques in the METRINO project, we have 2 materials, the citrate couple converting nanoparticles and the citrate capped iron oxide nanoparticles. And we already have the pre-characterization of the material. We bottled 100 to 150 units of the 2 materials. The homogeneity study by Sachs is already successfully finished, and currently we are doing the stability studies by Sachs and DLS as an additional technique. And once this is also hopefully successfully finished, we will distribute the material to the Sachs groups in the project to have a small interlaboratory comparison with this material. And finally, also the cytotoxicity of the two materials will be tested, so that at the end we should end with 2 reference test materials as standards for size and particle number concentration with known cytotoxicity, which can be used for measuring the nanoparticle uptake and distribution in single cells and tissues. And at the end, I would like to thank the organizers of Nanomed Europe for giving us the opportunity to present our project here, to all our partners in the project, and of course you for your attention.
We've made a lot of progress in the reference material, right? Thank you, Elina, so much. Now, please welcome Enrica Alasonati on stage. Despite she's Italian, she works for the LNE. LNE, I can do it. It's Laboratoire National de Metrology et d'Essai. Yeah, I'm better than for BAM. Enrica, you will talk about improving the analytical reliability through interlab comparison, right? The floor is yours. And
I will try.
Yeah, the slides are almost there.
Are you tired? Please stay with us. We still have 2 presentations and then the discussion.
If I'm still alive, you should all stay. Okay.
So, thank you very much, actually, that I'm here, because it's my first participation to this conference. So for me, it's a first full immersion in the nanomedicine world. And I would start with acknowledgement of all this fantastic stuff that we are working together, because we have a lot of people inside the project and also stakeholders. Some people are producers of the nano objects, and the other participated to the method development and to the interlaboratory comparison. So thank you really to this very nice stuff. Now, I will not go back to the objective, because Robin already explained it before, the METRINO project objective, but I would like just to make a focus on one more package that I am leading, the one dedicated to the fractionation strategies. And about that, what we did in the first step of the project was to develop a robust sample preparation method in the idea to measure this object also inside matrices, and so to study a little bit the behavior inside matrices, and with the final output to go to standardization of the nanomaterial analysis in biological matrices. So for doing that, we worked with different techniques. Basically, I would say that finally we focus quite a lot on field flow fractionation, and I will, in the next slides, go back just to the concept of field flow fractionation if you are not familiar with this kind of techniques. And we also compared with the size exclusion chromatography and with the complementary techniques like nanoparticle bio-Sachs. You have a specialist of Sachs inside this room, actually. And the idea was to use all these nanoparticles that have been produced inside the project to develop methods able to separate and characterize these objects in aqueous media and biological matrices. And we have here some criteria which come from the ISO, which is ISO dedicated to field flow fractionation techniques, where we would like to have recovery higher than 70%, and we also would like to obtain an uncertainty associated to the measurement for the size measurement lower than 5%. So, back to field flow fractionation. So this technique is actually a family of techniques which are is based on different physical principles, which the aim is to separate nanoparticles in a liquid media according to their diffusion coefficient and actually their size. So here we deal with the asymmetrical field flow fractionation. So in the big family is the one which is using as field separation only a cross flow, we call this, so a separation field which is a flow which is traversing our channel. I will not enter inside really on the specificity of the technique, but just keep in mind that this technique allows to separate nanoparticle, nano objects in function of the size, with small size coming eluting in the normal mode, eluting before, and a larger size eluting after. Then this kind of system is coupled to a lot of detectors, depending on what you have in your lab. Typically here I showed UV refractometer, we have a dynamic or a static light scattering, ICP-MS. And apart from ICP-MS, which is destructive, the other detectors are not destructive. So one of the power of this technique is that after you can collect fraction and add many other analyses of line, like PTA, SEM, single particle, ICP-MS, etc. So very powerful technique, and I need to explain just one technical point here, because in the intercomparison that you will see after, we talk about standard channel and fleet inlet channel, so I need to make the difference for you. What is focusing? Focusing is a step, important step that comes in the beginning of the technique, where you make in order that your sample is focalizing in the beginning of your channel. The channel is like the chromatographic column in chromatography. And so in such a way, you reduce the broadening and you allow your sample to use the whole channel to well separate. But there is also another kind of channel that has been developed in FFF, which is the fleet inlet channel. Here, the idea is different. So we have not this first step where we are stopping the sampling in the beginning of the channel, but we have a kind of, I would say, dynamic focusing, because we let the sample enter from a fleet here. And this allows to have a separation, but in more smooth way. And the interest to have this kind of channel is that we increase in such a way the sample recovery, because the step of focalization sometimes make losing part of your sample there. So we were interested also to really to compare the two kinds of channel in this work, and this is what we did. A rapid idea of the development in the project, but I will really pass this fast. So we applied this kind of technique to the different objects that Elina explained you before that were produced in the project. So for example, liposomes produced by Correlative, and what I want to say is that we developed different methods that were successful for this kind of separation and characterization. Another example is metal oxides. Here you have an example of characterization of iron oxide ferraspin produced by Nanopet Pharma. And finally, lipid nanoparticle. So lipid nanoparticle and liposomes, I would say that were the more mature method, ready for standardization. Actually, liposomes were already published as a standard for the ASTM, And we would like to go for a standard to the ASTM, also for a lipid nanoparticle. So we focus more effort on that part, and we propose an intercomparison on that part. And so initially we developed a little bit the method in the aim to have a method which is well suited to separate the lipid nanoparticle from the matrix. And it's what we did here. So you see that it's easier to take out a neo from the matrix when you use FFF, because you are not obliged to use invasive method like extraction from the matrix, but you can, with the technique online, already obtain this separation from the matrix. So we decided to go on with the validation process, and here we go for interlaboratory comparison. Yesterday evening, I had some discussion with people, and we realized actually that metrologists use a lot of words like usual words for us, but finally is not so speaking to the other people. So I decided to put back some definition, and I saw that also my colleagues did the same. So that's great. So it looks like intercomparison is something easy, but finally it's always better to remember the definition. So the idea of intercomparison is to organize an evaluation of the measurement on the same object or similar object that are distributed to laboratories in accordance with predetermined conditions. And we will go back after to this predetermined condition. And so here you can see that we have basically 3 types of intercomparison that can be proposed with different aims. One possibility is to use intercomparison to characterize and to give a reference or certified reference value to a material. We can use these for the evaluation of the measurement performances, or we can use these to evaluate the participant performance. And in this case, we talk about proficiency testing. So in this case, the intercomparison that we have organized was in the aim to evaluate the performances of the method. So we decided to do this on fresh prepared LMP material produced by Syntef and Correlative. These are aqueous batches of LMP on patro formulation, mRNA and siRNA pegylated, and they were prepared in January and distributed to the partner the 28th of January, so this here. The participants had 2 weeks to measure them. And in parallel, we started stability study covering the 2 weeks of the intercomparison, but also in the idea to study for the longer term the stability of this object at 4 degrees and minus 20 degrees. So this is still ongoing, and the results are in treatment. The participants were participants of the project, but we had also some stakeholders that joined us for the exercise. And one point which is very important here is that for FFF systems, we have 2 producers in the world, and the theory is the same. The practical instruments are slightly different, so there are specificity. And one of the point of this exercise was actually to develop a method which is able to be transferred to different equipment and which is working for different equipment. So we had here different configuration in the sense of free inlet and standard channel, as I explained before, and different equipment coming from Postnova and Wyatt companies. And then also we had, as participants, 2 partners that were studying by MADIALS and 2 partners studying making measurements by SACS. So before I told you, we will go back to the predetermined condition. So what does it mean? That means protocols. And when you organize intercomparison, actually this is something that you have to decide how you will be strict on that. Are you leaving the participants to use their homemade approach or not? In this case, we decided to fix as much as possible. Oh, it's finished. I'm speaking too much. So we decided to actually to fix as much as possible all these parameters because for people that use FFF, maybe they know that there are a lot of parameters for the separation, for the detection, and for data treatment. And so we, no. I don't need slides. But these are easy slides. For the result after, maybe, because... yeah, it's back. And so we decided to fix, so I will not go through the detail, but you have here all the main parameters. We decided to have 9 injections on 3 separate dates to try to cover, the final idea was really to cover a typical analysis that a routine laboratory is going to perform, taking inside all the uncertainty that you can have because you are changing the membrane, because you are working on several days. So we decided to cover this with also an analytical sequence that was imposed in this case with washing, blanks and everything, and which was the same for everyone. And last but not least, a well-defined data treatment. And this is always quite tricky. We had 1 million of hours of discussion about how to treat this and which was the more representative way, the more representative also of the way that people usually do in their laboratory. Finally, we found an agreement. Yes, here you have the definition of measurement. This is, I share this because we are used to speak about measurements in metrology. No one speaks about measurements. And so just to let you know that the measurements is the quantity that you want to measure. Okay. And so finally, you have the list here for the different detector of the size distribution average value that we decided to measure. And also we decided to measure in the full white at half maximum, which is in chromatography correspond to the half part of the peak, but also on the full peak to go through the entire size distribution. What is in red here is the results, concerning the results that I'm going to present you just right now. So it's just a part of the result, because the result, we collected the data just recently and the data treatment is still ongoing. So to have an idea of the results, overview of the results, here you have everything together for the first sample and the second sample, but all the different free inlet and standard channel configuration and system from the different producer, supplier. So what we can say is that first recovery was higher than 90% for all the participants. So this is very well, it fits well the requirement of the ISO. And on the other side, we have the size variation between the participants, which is, I would say, quite good. Because if you take in account all this variation, actually we are here to make an example, we are between 36 and 43 nanometer. That's really a good result for us. And then these are the same results, but just putting more the focus on the difference between the standard channel and the frit inlet, and you can easily see that there is a difference in the size, which is smaller for the standard channel. I will not go inside the detail because this is something that we are discussing why there's this difference. We have the impression that probably the separation is more performant with the standard channel than with the frit inlet. And we know that the recovery is usually better with the frit inlet than with the standard channel. So it's a kind of compromise to choose one configuration or the other one. And so here you have the average value, and we can see that there's in average difference which is significant. And then finally, we progressed to the data treatment, because the data treatment of the intercomparison will be performed according to the ISO 57252, which aim is to evaluate the repeatability and the reproducibility of the method. And so basically you will have from the data treatment the repeatability, the reproducibility, and also the interlaboratory variance. So all the values are correlated together. And we can also calculate a ratio which can give us an indication about how significant is the interlaboratory variance respect to the other contribution. So when we apply the ISO data treatment to the two different samples, what we do also from the statistical point of view is that we evaluate if all the laboratory can be considered there, or if there are some laboratory which we need statistically to exclude. This can be because they are farthest from the overall mean of the results, or because they are close in mean, but the individual values are quite spread off. So in this case, we noticed that we have excluded between 1 and 3 laboratories, depending on the samples. But then, okay, this is preliminary, so we have to check about this, because statistic has some, I mean, it's not really we apply that and that's the truth. So we have to discuss a little bit and evaluate the different tests that we can use. But for the moment, so these are the first results, and we observed a very good repeatability. So the repeatability is the small r that you see here. That means that for the size measurements, we are lower than 2%, and this is very good. And for the reproducibility, we have something between 5 and 7, I would say, and interlaboratory variance is between 5 and 7 again. So there is the ratio also is here. When the ratio is quite low, that means that interlaboratory variance is significant. So that means that if we have to find a problem there, it's more in the difference between the laboratory than in the repeatability of the laboratory itself. But in general, I would say that these are very, very good results. And finally, last but not least, we had, I remember that we had also SACS measurement there, and our SACS specialists were working also a little bit on interpreting these results using different models, and they were faced to a few challenges, because here actually we deal with quite polydispersed samples, and so this limits the analysis due to the lack of scattering filters. And also you see there are problems about inhomogeneous electron density contrast. And the other subject here of discussion is how to compare the size results with size result coming from DLS or multi-angle light scattering. So we are still working on that. Here we have preliminary results from our specialists, but most of this will come in the future. And I come with that to the conclusion. So We can see that multi-detector FFF is a well-established method for the characterization of lipid nanoparticles because we can separate from the matrix and also because we can provide repeatable and reproducible size distribution measurements. We can say, okay, just keep in mind that the full data treatment is ongoing. We are going to address all the measurements that you saw before, and also to address this comparison between the different techniques. And finally, we intend to use these results to contribute to the standardization, as I said before, activity within the ASTM E56, but also at the ISO level in the nanotechnology group TC229. So stay tuned about our results. Thank you very much.
It's better like that, yes. Leonardo very kindly submitted an abstract to the selection of the scientific committee and was selected. So metrology is also selected by the scientific committee of ETPN and NanoMed Spain. But you speak at your exact place, which is the METRINO project. And Leonardo is the specialist of imaging of nanoparticles in complex biological tissues and matrix. And he comes from INRiM. So as soon as I change the slide, Leonardo, the floor is yours. He was looking at me like, Alex, change the slide, please, and shut up. I did it.
Okay. Thank you very much, Alex, and everybody. I'm Leonardo Mortati from INRiM, the National Institute of Metrology of Italy. And I'm the work package leader of the last work package that treat about measurement of nanoparticles in biological tissue, phantom, and so it's, I would say, the practical way to see these nanoparticles, so really in action. So the idea is now I will show these preliminary results because it's still a work in progress, and it's a multimodal approach to measure the fate of these nanoparticles. So use different techniques and try to quantify, to localize the nanoparticles. So it's very important in nanoparticle studies to see where these nanoparticles go. So also to ensure safety, efficacy, and what happens to nanoparticles in vitro, in vivo, and so on. So these studies require detailed information and advanced characterization that sometimes not just one technique can solve. So in general, we need to detect these nanoparticles, to quantify, to localize where are the nanoparticles, to have a chemical characterization, and also to understand what the biological environmental surrounding the nanoparticle can influence the nature of the nanoparticles. So the idea is to combine different imaging techniques in order to have a correlative approach and to combine the strength of these different methods and have a more complete picture. So we have the imaging that allows to visualize the spatial distribution of the nanoparticles. And we have also spectroscopy or spectrometry techniques that can provide chemical information. In general, this correlative approach is made case by case, and this could lead to different drawbacks, like difficulties to compare results across different studies, some challenges in assessing data reliability, and uncertainty in measurement budgets. And these lead at the end to a slower clinical translation of nanomedicine. So in this presentation, I will show you these preliminary works that have the aim to establish a standardized protocol for nanoparticle analysis in biological samples. So we use two types of this list of nanoparticles, but the idea is to use all the nanoparticles that you saw in the previous presentation, so also lipidic nanoparticles, iron oxide, and so on. But in this case, I will show you just these two types of nanoparticles that are the upconversion nanoparticles and the hafnium oxide nanoparticles. The model that I will show you are, in this case, two different models. So 3D in vitro engineered tissue scaffold seeds with the A549 cells and liver mouse tissue. This work is made by different laboratories that are physically distant. So the idea is to really build this protocol, the workflow, so try to have also a feedback, because actually sometimes there are some practical issues to be solved. So the idea is obviously to use the same sample and circulate the same sample to the different laboratories. So in a way that everybody measure the same sample and correlate and compare the results. Of course, there are some non-destructive techniques and some destructive techniques, or mildly destructive techniques. So the first techniques obviously are the non-destructive techniques, in a way that the sample could be preserved from one lab to the other. So at the beginning, in general, we image the whole sample in a way to have a low magnification, because sometimes these samples are very big in terms of imaging, in the sense that are millimeter per millimeter or centimeter per centimeter. So they are very huge. And usually we use a wide field optical microscopy or other non-destructive techniques, like, for example, nonlinear optic microscopy, to have also a preliminary idea of the distribution of the nanoparticles. So not only where is the sample, but where are also the nanoparticles in the sample. So the idea is to use these first optical techniques like nonlinear optical microscopy, Raman, fluorescence, that could be actually interchangeable in the pipeline, because they don't give any, I'd say, modification of the sample. Later, to use scanning electron microscopy with the energy dispersion X-ray, because sometimes these techniques need a carbon coating that could interfere with the other techniques. So in order to have preserved always the sample, we put these techniques later. And then the destructive techniques, we can say, so the top seams and the laser ablation ICP-MS. If these techniques are used gently, just over a very superficial layer, actually this sample can be measured again. So they could come back in the pipeline, so to be remeasured and also to understand what is actually the effect of these techniques over the sample. So just to explain a little bit the techniques that are used in this presentation. So the nonlinear optical microscopy is a particular imaging techniques, optical techniques that use some pulsed laser. So we have some picosecond pulsed laser, and we can have different techniques simultaneously, exciting nonlinearly the sample. So we have coherent anti-Stokes Raman scattering that can provide chemical contrast. So it's a nonlinear Raman. We have like 6, 8 orders more intensity than spontaneous Raman, because we drive the molecule in an excited state. We can have a second harmonic generation, for example, to image collagen. So it's a label-free. These two are label-free techniques. And also we can have two-photon excitation fluorescence. So when we want to see fluorescent labels, like in this case, upconversion nanoparticles, we can use this two-photon excitation. So The idea is to analyze the same sample from different point of view, and we have the advantage to use label-free and label techniques, better confinement of the emission and the focal volume due to the nonlinear process that leads to sub-micrometer 3D resolution. We can have a better penetration, also hundreds of micrometer and the lower sample damage because we use near-infrared sources, and the low invasiveness and low sample perturbation. We can also measure living samples per day and make a 3D scanning of the sample. So the other techniques are scanning electron microscopy. In this case, we use electron beam to image the sample with a very high spatial resolution. We have up to 3 nanometers resolution. The idea is also to couple these techniques with the energy dispersive X-ray spectroscopy that can give a precise identification of the elemental composition of the sample. So for this reason is also label-free imaging of inorganic nanoparticles, so above all metallic components. We can have also a very high contrast of the surface morphology with atomic composition. And if some additional heavy metal staining is put in the sample, they can also have ultrastructural detail of the biological context. It's not possible to image live samples, and also the surface that can be measured is not very wide, so it's limited. The other technique is laser ablation, inductively coupled plasma time of flight mass spectroscopy. So in this case, there is a laser beam that can ablate the sample, and part of these compounds that compose the sample can be accelerated in this chamber, and they can be analyzed in a way that they can be resolved from the mass of the compounds. This is a very high efficiency technique, and it is also a reference technique because they can have quantitative elemental maps, so it's a very interesting techniques. They don't need to prepare the sample. They can also image a living sample. Of course, this is a bit more destructive because we have a laser ablation of the sample. So actually, the first layers of the sample are ablated. It's a label-free detection of metallic nanoparticles. So it's very difficult. It's impossible to see the organic material. And the spatial resolution is around 2 micrometers. So for this kind of comparison, it's very nice because they can measure the metallic compounds and also the hafnium. So it's very high sensitivity, and there are no spectral interference between these elements. The other techniques that is also very nice is time of flight secondary ion mass spectroscopy. In this case, we have a primary ion beam that create secondary ions. These are measured with the mass spectroscopy, so it is possible also to see fragment of protein, so also organic material. So it's very nice because in this case we can have molecular information, not just the elemental analysis. So we can also have an idea if there are some nanoparticles, for example, degradation, oxidation, if happens something to the nanoparticles. And we can have a spatial distribution of molecular spaces in the image. So we have a lateral resolution sub-micrometer, so 0.2 micrometer. There is no labeling required. the sensitivity of the surface is very high, so 1 nanometer, and usually the ion beams is made by argon ion, and it can give a very high 3D analysis of the sample. So the idea is to compare the data obtained from each techniques and build a protocol that is optimized for this data integration in order to have the nanoparticle distribution within the tissue, the chemical identity and the speciation, and the comparison of primary and indirect methods. Because one of the lacks of these techniques is that, for example, fluorescence is not a reference and primary methods instead of, for example, laser ablation ICP-MS. So the idea is to have also a correlation of the spatial resolution and the sensitivity of each techniques. And of course, try to have a feedback if this workflow can work, actually. So the two kind of sample are, one is OCT embedded 3D engineered tissue, seeded with these tumoral cells and spiked with these upconversion nanoparticles. So here you can see the wide field image of the sample. You have to imagine that this is millimeters, like 8 millimeters per 3-4 millimeters. So it's a quite a big sample. So in this pipeline, we have did a preliminary imaging of the whole 3D section. So the measurement was made in 3D with the nonlinear optical microscopy, localizing the upconversion nanoparticle with 2-photon excitation fluorescence and the collagen by second harmonic generation. Then there are some region of interest selected by the technique of Tom Sims, and also other selected with laser ablation ICP-MS. So here you can see the different regions. So this is the big image that is a very high resolution. You can see it's like 6 millimeter per 2.5, but we are talking about a million of pixels. So this is just some region that were selected, and so I will show this that is measured by all the 3 techniques. So here you can see in red the protein, in green the upconversion nanoparticles, and in blue the silicas of the glass. Here with the nonlinear optical microscopy, in blue the collagen, in green the upconversion nanoparticles, and here with the laser ablation ICP-MS, the tissue and the ytterbium element. And here you can see the overlap of the techniques. And so there is a good matching between the techniques. Of course, the resolution is not the same. So, for example, Top Sims is very high resolution in comparison to the other. And also we did the same work, but with these hafnium oxide that are not fluorescent. So in this case, we use SAM EDX, is a paraffin embedded ex vivo liver mouse tissue. And also in this case, we have a super image took with scanning electron microscopy and some region of interest that are chosen. So the results are here. So here you can see the hafnium took by the EDX, the hafnium oxide from Top Sims, and here the hafnium from laser ICP-MS. Also in this case, we can see the overlap. So there is a good matching between the different techniques. Here it's just another image of Top Sims where you can see that it's possible to see also the nuclei of the cells, the cell material of the protein, and determine where are the nanoparticles itself. So the idea is with this work to lay a groundwork for a standardized protocol, and validated through an interlaboratory comparison. In this case, in the frame of the METRINO project, but in the future also open to other laboratories. And this standardization should improve the data reliability and reproducibility, and also facilitate the comparison of results across the studies in the community, scientific community, and this could accelerate the clinical translation of nanomedicine. So I would like to thank all, of course, Nanomedicine to host this talk, but also all the colleagues that participate and METRINO, of course. And of course, also you that listen to the talk. Thank you.
I'm perfectly aware that you're exhausted. We are all exhausted. But I mean, we took planes and burned some carbon to be here. So let's take 15 minutes of your time, not anymore, just to try to have this panel discussion and to make it very useful. Can I ask all the speakers to come please on stage while I present a very important thing. Yeah, you can't see it if I'm in front. I'm perfectly aware. I'm arriving in 10 seconds. Yes. Yes, please. Okay. Okay, so I learned after 10 years of ETPN with low resources that very simple things works best. So I would love for you, it's possible to show, please, the video still. I'm aware it's a very special thing. Yeah, you would not believe how happy I would be if some of you at least scan this code and follow on LinkedIn METRINO for the updates of these guys in the coming months. By the way, please have a seat now, for real. The mic are supposed to work exactly. You could open it. Please have a seat. First, congrats, guys. I mean, I'm within the project and I'm amazed about the result that I saw today. Big progress on all the key pillars, I would say, of METRINO. Maybe Elina, let's start by you. You've shown a diversity of potential reference materials. I wish to do a survey about who's aware about the difference between certified reference material, but I won't do that now. No, you've shown a very diverse type of reference materials. Would it be for the material or for the property that we certify, right? In your opinion, what would be the one that would have the biggest impact and the most easy impact to deliver among all these potential reference materials? The most impactful one and the most easy to take up by the community?
Well, here I think that the materials synthesized in the hot temperature methods, which can be synthesized in the biggest quantities, would be the material that we would be at the end able really to release from the project with reference as a reference material for defined properties. And I also think that we will have some very nice results on the lipid and liposome nanoparticles.
Who wants to comment on that? Enrica, Robin, about this diversity of possibilities of reference materials, or Robin?
Yeah. Yeah, I think I completely agree with the easiest one. I think the most impactful would be like lipid nanoparticle reference materials, as I think this is something which people are looking on. There the problem is like the patterns on the different formulations, because I told you that we are looking for like reference materials which are similar to the products on the market, and there's a huge jungle of patterns which makes it really difficult to produce something and to bring something like to commercialize something which is like similar to the products on the market.
Would like to ask the question to you. Yeah, I mean, first a question by Adriele, indeed. I mean, hopefully.
I'm setting up a standard. Huh?
Yeah, please.
No, very much appreciated. Thank you very much. I think I might have a suggestion directly to what Alex was saying. Why don't you partner with somebody that is trying to do some clinical development and assist them in characterizing the material as a reference? So your selection goes, which one is the low-hanging fruit to lend the reference material to somebody that is trying to get into the clinic?
Close to there, but not there. Right.
Without shouting. So that could bridge the two. So you see the applicability, you don't need to produce tons, and then in the meantime, you can produce the large amount for the interlaboratory study. But at the same time, you're collecting data that it shows you that your reference material is properly characterized as you presented across the 4 different platforms. It's a suggestion.
Any comments on the suggestion from Adriele? It's not mandatory.
Yeah, it's a good one if you have like a connection to where.
You have Alex.
Yeah.
Alex is the person that solves all the problem. I mean,
yeah, indeed.
Within ETPN, you can post out a request on
Absolutely.
Somebody, and this is within your right, because as METRINO contribute to ETPN, raise a question here or bring it to discussion across the forum tomorrow.
You know what? Marco Monopoli left the room because he had a meeting and he said, from what I saw, I need to make a comparison of what I do in my lab with them. So I would like to get in contact. So the purpose of inviting METRINO within the Nanomed community is to start this connection and to start this channel.
As many of us, we are looking for reference material. I mean, personally, I'm looking at it. We have one of our sponsors at the conference looking for material to use in their development of instrumentation. I don't want to point the finger to anybody. They are
so
Irish.
you're not pointing towards Darren from Marava Labs, right?
They are Irish
no, you're not.
as well. So I don't know. There's a lot of this comparison.
Adriele, if I may, maybe a question to you guys. What would be the criteria that would encourage you to use these kind of materials even before official certification, while we are testing them? What would be the criteria to you and I mean to all of you? What would be the most appealing thing that would encourage you to use our materials? Does someone wants to answer? Maybe Darren. That was totally for you, in fact. But
sorry, Darren. I don't want to put you on the spot.
Thanks a lot. No, it's a good question. I think, I mean, what we do is specifically is trying to measure RNA payload in LNPs, in intact LNPs, which is different to ribogram where you have to break it open and what have you. And the biggest challenge, actually, demonstrating that technology is probably that nobody really knows how much RNA there is. So, you know, when you work it backwards, usually the way that someone's worked out how much RNA they put in was they measured it in nanodrop. And so, as far as I can work out, you're measuring nanodrops of things. And it all comes down to some UV phase absorption coefficient. So when you're trying to look at things like accuracy and precision and repeatability and so on, it's really hard to know what you're starting with, and particularly when you're working with different end users who have all got different samples, all prepared and measured in different ways. And you're going to then get compared with a technique like ribogram, which is known to have lots of problems and lots of sample measurement variability. So you've got a technique which is presumably a lot more precise, which is our technique, but you can't compare it with something like ribogram, which is the standard because it's all over the place. And so you're comparing one thing with, you know, apples and oranges very much. If there's no gold standard reference technique, then it's almost impossible to really know who's better because you're just comparing multiple different techniques. That I think is the biggest problem, that the lack of a gold standard that everyone can use means that you're comparing different things with each other. There's no kind of level playing field.
I'm sure you want to know more, and you will know more because we have a sponsor talk from Marava Labs just after the keynote tomorrow morning, by the way. Oh, please.
Yeah, basically the point is, I think I might agree, the ideal standard is a material that can have some different characteristics that can address the limits of all the techniques and also be stable, easy to be measured and so on. So the ideal maybe is not really the biological, it's a paradox, but it's not really the biological application, but it's something that helps to calibrate the techniques in order to have this traceability.
We have that. We're using this traceable, many of us, all the time. So we are beyond that. We don't need to have an absolute readout. We need to have a readout. And we need to start somewhere. And what Darren was addressing is the RNA loading in NMPs as an example. It doesn't need to be the total calibrators, because otherwise we never end. But in the LNP, as you know yourself, because you synthesize it, there's only some reference material produced by the Canadian. They're for temperature control. They're not for load. One example, you mentioned about the USPO, the non-linear particles for measurement, which are very fascinating, but it's where you can start distributing and getting feedback on your measurement in an interlaboratory exchange. Because if you continuously produce the material without getting out to hear what they want to do, you might be too late to distribute your material. You see where I'm coming? So my question, which was a much simpler question, is what's the next strategy for all your beautiful materials? Because you said it's 36 months, isn't it, the project? So in interlaboratory, by my own previous experience, took us 5 years. You don't have that time.
Enrica, please. Or Robin, both.
No. Yeah, I mean, you touch many different points, so it's hard to answer. I mean, interlaboratory comparison can take some time, depends where it's organized and which is the aim, how many participants, etc. For example, in the metrology world, it's right that it takes like 3 years. Before that, we agree on the final statistical treatment, and meanwhile, the research world is far away. So that's one point. But for other comparison, especially small comparison like this one, this is more easy to organize and more fit for purpose. And in the case of METRINO, this was also related to the previous work that was already been done by NIST and SINTEF on LMPs and related to ASTM. So the need was already there, actually. And we are using in the project to go on this topic. Then maybe there are a few stuff that I have not completely understood, and we can discuss a little bit also after. But I was thinking to one point that for us is important, and where I have the impression that as metrologists we are a little bit lost, is the matrix around the object. The matrix should be relevant for the study that you are really doing, the clinical study, etc. And it is not really there. I think we really need your input about what you need, because you are studying what this particle, how they behave in the human body for the use that we want to have for them. We need to know what is really relevant for you, which kind of matrices, but taking in account that matrices is difficult, and if you wanted to have SOP, if you want to have a measurement that you trust in, you should start from something easier and then go to something more complicated. We stop here.
Robin, you wanted to comment on that? If not,
yes. No, you.
No, okay. No,
I have a.
Oh, please.
Yeah, I have a little question. So I have plenty of questions. But
Let's start by the first 10 ones.
No, regarding the matrix, okay. sometimes with the nanoparticle we develop, we want to have an interaction with the matrix, or why not no interaction with the matrix? And do you think it has an impact or not?
Yes.
But finally, did you propose different SOPs for this or not?
We are studying, it depends from the object, it depends from the matrix. With this lipid nanoparticle, for example, it was easier stuff. I mean, it's in the results also that I showed you before, you see that even when we tested with different biological matrix, basically or simulated matrix, not real matrix. But we see that we can separate, even online, the object from the matrix, and we find back the size distribution.
Yes, but sometimes it's impossible to
yeah,
separate.
it's a different story, completely different story for metal oxide, for example. Here, depending on the matrix, you have different behavior, you lose your compounds, you have agglomeration, and so in this sense, also really, we have a lot of questions because we wonder, okay, but we would like to do a work for harmonization of the measurement, but we cannot. I mean, I'm sure that you are doing a lot of work on that because you want to, for example, for people that produce the material, you want to validate this for your purpose. So in this sense, we needed to have really the interaction to understand where you need our support for the work that you are doing.
Enrica, how open will we be to include these people in the comparison? I mean, is it just for METRINO partners, or do you think that there will be space to include their data, their material to test in the...
You are talking about to make a comparison?
Yes.
And
or to initiate it.
You know, it's a budget problem. That's all. It's just in this sense, because from the strategy point of view, of course, that this is really great. Then the point is that with maybe with metal oxide, it's easier because they are stable at ambient temperature. But for liposomes and LMPs, especially LMPs, they are quite fragile. So it's just a story of sending the sample and everything. So we should see what is possible to do there. But interaction, this is no cost, and this is great.
Okay. If no question, then I'd like to move to your topic, Leonardo, because time flies and we won't solve all the interaction problems right now, right? I think we should have more space to do that. But what you've shown is really advanced techniques for complex matrix. And my question is how these people can benefit from that. I mean, do you think that they need only collaboration, or they can change a little bit their practice in the lab to be more ready so that you can collaborate, have data available? So to make it more simple, how accessible these results and these methods are? Because we all want to know how the nanoparticles behave in complex biological media, but is it just for super specialists, or do you think it's open for people?
I think actually it's open. The idea I showed just some advanced technique, but is also to involve more, I would say, usual techniques. The advanced techniques can give some accuracy, precision that maybe the normal techniques doesn't have, but the idea is to create a procedure, a protocol that can give this traceability in the ideal expectation to the techniques that are widely used. So the idea is really to give this bridge between different techniques that can have different metrological characteristics in order to give this traceability, or try to have like a traceability also to fluorescence microscopy, for example, that is widely used. So that's the point. Try to have a sample that is a biological sample with a model, and try to give a procedure where it is possible to calibrate the different techniques. So it's really making simple something that is very complicated. Okay. So this is the starting, because it doesn't exist. So we are trying to solve a problem that is very, very complicated, actually. So we need actually more, the idea is to have the feedback and also the collaboration external to METRINO, maybe later of METRINO, we have to see. But the idea is to prepare the community to have a comparison that is open to others.
So in the second part of METRINO, please, Robin, come on.
Yeah, like the biggest advantage of a project like METRINO is that we have like, we are doing this kind of wild ILCs, so we can do the interlaboratory comparison like to our rules, so we can invite whoever we want and to participate. So there are like other ILCs like for more on the expert level, for example, for these RNA encapsulation, RNA amount, there's like from the NIST, some pilot study, which is like, they are working on this reference materials, but it is at the moment quite closed, so only like for metrology institutes where you can't participate, but in projects like METRINO, this is like very open, so we can get in touch, and if you are like interested in joining such ILCs. And
we had an impossible task that was to do this panel in 15 minutes. In fact, it's impossible. We need more time, but I feel that I'm more motivated than ever to create more connection between what we do and what was the nanomedicine stakeholders do. So we're going to think about, maybe it's an online workshop or webinar on a very specific topic that you can participate even after that. But it's clear that we need more of your inputs, and you need to understand more how to use the knowledge and the materials that will be produced. But maybe just to finish, if you have 2 minutes of extra stamina, I have a very simple question for you. You've heard about the level of expertise within the project. It's a tough project. There are tons of things we can do, and we have tons of possible group of stakeholders that we can impact. And on top of that, we also try to change the mind of the regulatory and the standardization body. So these are our heroes, right? We should consider them as pioneers and heroes. But still, if you had one simple thing that you think from this kind of expertise would change your daily practice, what would it be? Like, I would really like to know what is the right method for this, or if I had just a simple material that I can rely on that. I mean, simple ideas that would really change your work in terms of standardization. Any ideas would be greatly appreciated.
Thanks. So it's more towards the regulator. So the standards are fantastic, and I think the community needs the standards. Different analytical platforms coming and using those standards is going to give us harmonization. In terms of what the regulator will ask somebody as you take it towards them, my experience is they don't care as long as the analytical technique is going to prove that your batch-to-batch manufacture is consistent for release. So I suppose what I'd want to see is you working to convince the regulators that those techniques, new and existing, are the ones that they'll look at and think, yes, this is giving me the confidence as I assess an approval for release.
Can we realistically have an impact on that during the time of the project? Be honest.
So I guess the question is, how close are you to the regulators?
No, I think we're not that close to the regulators like we wish to be.
Yeah. It's not that easy, right?
Yeah.
Okay, but at least we have a clear need. Another idea, I mean, something that in your daily practice, please, come on. No, you need to speak up because of the record, it's better. Only Adriele is on the record without mic. You don't shout, you.
It's not that I think it's a comment. Open the mic. It's a two-step process. So it's not an idea, but it's a comment. Yes. I don't have the apparatus you speak about in my laboratory, or I don't have a daily access to this apparatus. Exactly. So it will be very complicated for me to apply your protocol, but I'm so far from the clinic with my research activity, but it will be really complicated for me to apply your protocol, your standard procedure in my research activity. So what I have to do?
And if I may, what should we expect until the end? I mean,
yeah, we are not supposed to have all the techniques and all standard operative procedure and all the material for all the measurements. this is too much for sure. But we are trying to develop a kind of a structure, metrological structure, I would say. So it depends, for example, which is the parameter that you are measuring. Maybe you have another techniques in your lab which is giving the same parameters. You can link. I mean, this is common in metrology, that, for example, you make a calibration of an instrument by, there are several ways to do this. So it's once per year, for example, or something like this. This you can do only if there is an existing structure. And this is what we would like to build. And for that we need reference material, and we need also really the measurement capability in the metrological world, because otherwise we cannot offer this kind of service. So this is what we would like to build up a little bit, little by little, and that's why your input is important. And I would say another thing is that we are talking about METRINO project. METRINO is going to finish next year, but metrology still go on in this sense. So we know that nanomedicine is strategically important, and there will be other projects. And just to inform you, for example, this year there was the health call, and we have other colleagues proposing new topics. So this is really something which is, we are in the real beginning. And so, yeah, that's why it's important, this kind of communication.
Yeah. Oh, you.
Just one very simple thing. Like back in the 90s, you know, people used to use DLS all the time for particle sizing, and it was well understood and known then that if you want an accurate instrument-independent measurement, you extrapolate to zero wave vector, 0Q, because every instrument has a different wavelength laser and a different measurement angle. Therefore, the results that you get are completely instrument dependent, and they can't be compared directly with each other. The way you get around that is you extrapolate to zero wave vector, then you get an independent measurement. So CQAs should be Z average diameter polydispersity as extrapolated to 0Q or zero wave vector.
Yeah, and it's all about practical guidance, right, for the people and how we can do that. Well, Adriele, yes, let's kill the people a little bit more.
So, absolutely. No, I think you mentioned yourself, you started part of your manufacturing for metrological sample through the ASTM work that it was done by Syntef. So maybe that could be the trajectory that you could follow. ASTM is just only the entry point in the regulatory space, connected to what Stephen Conlon was saying. And you go towards the ISO. So you bring from the ASTM as a trajectory of METRINO continuing through the metrology in the ISO, because that's where you actually set up the metrological standard. And through that one, you initiate a working group, you initiate a dialogue between the already METRINO partner, which are already referenced in the national countries for metrological assessment. And this is similar to what Caterina Minelli did on MPL on the Bahamas. So maybe that could be the pattern. And we are already involved into part of the work with Syntef and NIST and everything. But that is, you know, it's doable because all your metrological organizations have already that content in the working group in ISO.
Yeah, but this is ongoing, really, because there were some changes actually in the leading of the CEN and the ISO also at the UK level. And actually, one of our colleague, Dorota Barczak from LGC, which is not here, but it's part of the project, is going to bring probably in the ISO this FFF method and actually the result of this intercomparison. And the idea was really because we are going to be in parallel with the ASTM. The focus is at the level of protocol. There are differences, I mean, in the way that is, so it's good. It's not overlapping, it's parallel, I would say. So that's for sure. Yeah, thank you for your remark.
I'm sure that after a good night and some extra coffee in the morning, you can find these people. They will stay for the full conference. I mean, at least almost the full conference. I would really like to thank them first for the presentation, and thank you for staying very late. We are late, I know, but it's so precious for us. And my message after that is that we need to organize key workshops on a very precise topic to get you involved in an easier way. But they needed first to gather the data and to start the process. Now we are ready to hear you guys, and I guess that would be what we will do in the next part of METRINO. Again, thank you. Sorry for being late, and tomorrow, fun day, you will see.
And thank you, Alex, for allowing all this.
It's strange to conclude, thank you for this day, and tomorrow it's going to be fun, but it's because it's going to be super fun tomorrow. We have the gala dinners, I mean.